Okay, just a review of hypertonic and isotonic again. So essentially, if you have a lower concentration here, this is a selectively permeable membrane. So our sugar molecules can't go back and forth. This is a higher concentration. What is water going to do? Water wants to dilute things. So water is going to move over here. So again, it's going to end up being an isotonic solution because the water moved to dilute this section. That is also going to happen in your blood. If you put this in essentially in a hypotonic solution, the water is going to rush into this blood cell to try and dilute it, and it will burst. If you put it in a hypertonic, the water is going to come out to try and dilute it, and it will shrivel up. You really only see this effect on plants when it is in a hypertonic solution, where the water is lost from the plant and the plant will shrivel up. Otherwise, you don't really notice it as much. Okay, so there again, the sugar molecules will diffuse through the tube until equilibrium is reduced. If you didn't have a semipermeable membrane, if you have a semipermeable membrane, the water will move. So that's the answer to that one. Okay, the laws of thermodynamics, always good times. The first law of thermodynamics states that energy is either created or destroyed. If this is true, where has the energy of the diver gone? So this is that picture of the diver who's on top of a, where's the diver picture? So think of a skater. I like to think of a skater on top of a mountain, but they're talking about this picture. So where does the energy go? To the heat. So essentially, this whole process is creating heat, and there's going to be heat, especially when heat hits this water, depending on how you hit the water. And that doesn't look like that's going to be a great dive. He might pull it out in the end, but I don't think so. So your energy is going to be lost as heat, and that's going to increase your entropy or your disorder. So essentially, this, remember, started with, can we engineer enzymes? What is the likely reason for the ability of engineered enzymes to break down different sugars? The reason is that mutations in the gene cause change in the enzyme's active sites. Changes in the shape of the active sites could possibly allow the enzyme to recognize and bind to different sugars. So that's how we're genetically engineering enzymes. And I think we do have some phagocytosis that I don't think I got to. So I can show you that real quick, and then we can be done. So essentially, a way of active transport, this is maybe the most important one. This is your sodium-potassium pump. So I'll just play that for you because it's actually a really good one.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy for ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein.
ATP, so it's active, it uses energy.
Causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
So honestly, active transport with your sodium-potassium pump may be one of the most important things that is going on in your body right now. Essentially, the active transport means it's moving it against the concentration gradient. It's shoving all the people in the closet, and that takes ATP. So I like endocytosis and exocytosis because I think it's just cool. There's a lower concentration gradient. Sometimes this can just be used with an active transport channel, but mostly it's the sodium-potassium pump. Okay, exocytosis is when the cell kind of wraps around it, makes a vesicle, and just sends it out. It's the movement of material out of the cytoplasm via cell membranes. This is what it looks like.
Endocytosis is the movement of materials into a cell via membranous vesicles.
You want to guess what exocytosis is? The opposite.
Exocytosis is the movement of Endocytosis is the movement of intracellular vesicles to the plasma membrane, where they fuse with the membrane and release their contents into the surrounding fluid. This process occurs predominantly in secretory cells, such as mucus-producing cells or pancreatic cells, that secrete enzymes into the digestive tract.
It's also how your tear ducts work. So basically, if it's outside the cell, it will fuse with the cell, and then it will just spew out the contents of your tears or your glands of some kind. Endocytosis takes it inside the cell, exocytosis takes it outside of the cell, and phagocytosis basically engulfs a particle and packages it within a food vesicle. So you have endo, phagio, and exocytosis. You have three kinds. Here's phagocytosis.
In endocytosis, membranes invaginate or pinch in to form a vesicle, moving the enclosed materials inside the cell. This process can take different forms, each involving its own specific cell machinery. In phagocytosis, or cell eating, the cell engulfs debris, bacteria, or other sizable objects. Phagocytosis occurs in specialized cells called phagocytes, which include macrophages, neutrophils, and other white blood cells. Invagination produces a vacuole, which usually fuses with one or more lysosomes containing hydrolytic enzyme.
So there is your phagocytosis and your endocytosis. Again, phospholipid bilayers are key to your simple membranes. It allows things to come in and out. And that is essentially the chapter. Here is a membrane transport. Here's diffusion just from high to low. Here's facilitated diffusion through this membrane. Here is just water can come in and out. And here is active transport, where you really have to have ATP in order to move from a area of low concentration to high concentration. Okay, see you next week.